Welcome to another screencast where this time we'll be discussing the structure of chromosome and dealing with here the levels of chromatin packing specifically in eukaryotic cells here. So from previous screencasts and biology classes, you understand that the DNA molecule is a double-stranded DNA molecule. It's also described as a double helix in structure. But then all the other times you hear this DNA or this genetic material described as a chromosome and sort of diagrammed like this here, and you have this from a poster that's talking about all the different names of the genes and where they're located, say here on chromosome number 9, right? So we have this chromosome term, but then we want to look at how is that chromosome term related to this thing, DNA here. So if we take a look at the length of a chromosome in a typical human eukaryotic cell, right, there's about, on average, 1.5 times 10 to the 8th base pairs long. Again, this is the mean or the average of all of these chromosomes right here. So if you took one of these chromosomes on average, right, and spread it out, it'd be about 4 centimeters long, which is greater than thousands of times the diameter of an individual eukaryotic cell. So the question is then, you have in the human cell, you have 23 chromosomes, 23 different ones. So if just one of them is this long and has these characteristics to it, you're gonna times that by 45 more, and you got to get all of this material into this nucleus right here, which is indeed smaller than the cell. So the question is, how do you get all these linear chromosomes into this nucleus of the cell? Well, the answer is that the chromosome, that the DNA molecule is packed, right? It's condensed, right? So here we have packing as we go from left to right, or we say that the DNA is condensing as we go from left to right. And then as we go from right to left, we talk about the chromosome unpacking or being decondensed, right? And so it turns out that this DNA molecule, the double helix, is a chromosome, and the chromosome is the DNA. But the chromosome was first discovered back in the you could see this in the late 1800s with crude microscopes and the staining techniques, right? So they saw these colored bodies just in the cell, and as microscopes got better and technology got better and better and better, we could then use electron microscopes over here to see all the way down to this 2 nanometer size and diameter. So this is just the DNA double helix made of nucleotides. But then to get that thing packed up, it turns out that DNA is wrapped around this set of proteins called histone proteins. So really a chromosome is DNA and a set of proteins, right, that DNA is associated with. So then as our understanding of what the chromosome actually was, what the DNA molecule actually was, we came up with another term that is described as being chromatin, right? And this chromatin can also be sort of loosely packed together, in which case we refer to it as euchromatin, or it could be a little more tightly packed closer to this chromosome side over here, in which case we call it heterochromatin, right? Because it wouldn't be biology without having about 3 or 4 different names for kind of the same thing here. So let's talk about this packaging here and how this actually occurs. So as I said, there are these group of histone proteins, and it turns out that these histone proteins are relatively positively charged, right? And these histone proteins group together in sets of 8, or these octamers, and then the negatively charged DNA molecule will then interact with those cores, right, to tightly bond around those octamers, and you can see here that the DNA molecule is looping around these octamers twice. That DNA then it would continue over to here, right? You'd have this double helix continuing over to the next group of positively charged histone proteins, and this conglomeration thing here is referred to as a nucleosome. So these nucleosomes, these bodies within the nucleus here, consist of these DNA coiled around the histone proteins. And then interspersed between that then are these stretches of DNA that are wrapped around the histones. And this is this linker DNA. And it's this linker DNA that is actually then available to be replicated. It's unpacked. It's just DNA, the double helix. It can be involved in replication, but then also in gene expression, the first step here in transcription. So we see that this winding of the DNA around histone proteins can play sort of a regulatory role in what the DNA is doing or how the DNA is being accessed inside the nucleus. And there's more on that later. So a little bit more on how this packing occurs. Well, we see that these histone proteins here have this positively charged core, but then there's this tail coming off of here, and these are the N-terminus parts of the amino acid sequence. And these are available for chemical modification. And the three chemical modifications that occur is you can put methyl groups on there. So this would be methylating. Right? So this is a methyl group. This is an acetyl group or an acetyl group. Right? And so you can acetylate or acetylate these tails. And then this is a phosphoryl group down here. And so you can phosphorylate those things as well. So this combination of methylating and acetylating and phosphorylating these tails over here will play different roles in that packing of the chromosome. So if we look at this here, we can see that these acetylated histones are right here, where these green dots are the acetyl groups. Right, so this is the acetyl group right here. And when the histones get acetylated, we see that that the DNA sort of unpacks a little bit more, and this is the euchromatin form. But when we deacetylate or unacetylate these histones, we see that these tails will then interact with one another, right, of all the histone proteins, and bind that up a little bit more into a heterochromatin form. Right? So we can then, of course, reverse these here. And what does this acetylating and deacetylating, of course, are a series of enzymes that are specific for sticking these acetyl groups on or taking the acetyl groups off, as the case may be. And again, these enzymes, the activity of these enzymes plays a key role in regulating the expression of genes and the replication of that DNA. So these are the primary chemical modifications, but we also want to include here that we just mentioned that you can phosphorylate and dephosphorylate these histone proteins as well, and the dephosphorylating and the phosphorylating also will play a role in making it either euchromatin or heterochromatin. There are then also methyl groups, so when you demethylate the histones and methylate them, you again get the same sort of packing and unpacking here, right? So acetylating, phosphorylating, and demethylating the histones gets you this euchromatin, right? And then to make heterochromatin, to pack it up a little bit tighter, you take the acetyl groups off, you take the phosphate groups off, and then you methylate those histone proteins. Right? One final note here, that the DNA can also be methylated or demethylated. When you demethylate the DNA, demethylate the DNA. This also makes it unwind, the DNA unwind from the histones a little bit more. And when you methylate the DNA, that makes the DNA also bind up a little bit better, and you get this heterochromatin. So methyl groups, acetyl groups, phosphate groups play a role in which form that DNA is in, either the euchromatin or the heterochromatin form. And lastly, in recent years, this is a picture from Scientific American, and I forget the issue. I should really know that, but Scientific American article that I subscribe to, and I noticed here that they had this diagram, and it's this architecture of a chromosome. And down here, what was interesting is that in the last 15 years, advanced microscopy has given to the live a long-standing view that chromosomes sit higgledy-piggledy in the cell nucleus, like cooked spaghetti in a bowl. This image individually colors the chromosomes in the nucleus of a human fibroblast cell. So what basically they're saying is that, like, all of this DNA and all of this chromatin, yes, it's tightly packed up in there, but then it's not just thrown randomly into the nucleus. It turns out that some of these chromosomes, this chromatin spends its time in different areas of the nucleus, right? That some chromosomes hang out near the edge, some chromosomes hang out near the middle, some chromosomes are sort of always in this part of the nucleus over here as compared to other chromosomes which are in this part of the nucleus over here. And not only that, but they spend different times in different forms, going from euchromatin to heterochromatin, right? And there are certain areas of the nucleus where, when the chromosomes are being replicated or in euchromatin form are being expressed, genes are being expressed from them, that they move to different areas of the nucleus, right? So the more we look, the more we see that actually it is not random, that there is some sort of regulatory nature to the structure of these chromosomes. So just memorizing this stuff and knowing the packing terminology and how it packs and how it unpacks isn't really just random useless information. It turns out that it's actually meaningful, like how that packs, where that DNA is in the nucleus at different times throughout the cell cycle, turns out to be actually meaningful. So very interesting stuff there, at least I think. And finally, we just do want to mention that in prokaryotes, they have typically one chromosome. The genomes are a lot smaller. They're not membrane bound. But in this nucleoid region, we see DNA as well, and it will also be packed around proteins and folded. And again, used to think just sort of like randomly folded up in there, but turns out that it might actually be not as random as we thought, and that the folding patterns might actually have some sort of meaning to them. So, all right, well, I hope that is helpful in sort of helping you describe this levels of chromatin packing in specifically in eukaryotic cells, and a little bit about how that happens and why that might be important. Okay, thanks for listening, and bring your questions to class.